All right, what we have here is our taste and smell. And so first for taste, how this works is we have two kind of different systems. We have one for salt. Oh, didn't mean to do that. One for salt and one for sour. And how that works is we have, for salt, we have sodium that comes in, it creates a bong, and that gets this to go, it gets voltage calcium to release these snare proteins, and it goes and creates a neurotransmitter that then creates an action potential. And then we have it for our sour, which is just a proton that comes through this ENaC. And this one's different because it blocks the K+, the potassium leak channels. And what that does is it makes it depolarize even more. And so it just makes a more intense response. Then we have bitter, sweet, and umami. Umami is Japanese for delicious. And how this works is there's a ligand that comes and it binds right here to that G protein. The alpha subunit will then go activate the phospholipase, and then it will use IP3 to then go to the RYR, let calcium out here, and then stimulate the neurotransmitter to go into the synaptic left that way. And so the main difference is these two are ionotropic and these are metabotropic because they use the G protein, and these use the ion channels. And these are kind of the subsequent chemicals that cause them to give a certain signal. And so taste buds are really cool because they replace themselves every 10 days. Smell actually does it every 2 months. So they both are replaced, which is kind of a unique thing about them. When we taste spicy, those are actually nociceptors, which are pain. And then we have the nerves. We have the facial, glossopharyngeal, and the vagus. Those are all part of taste. Smell is what I said right here, that every 2 months, and they routinely die and are replaced. These receptors are concentrated in a place called the olfactory epithelium. Okay, right, that's right here. And so it has all those nerves. And so what happens is you smell something, that's the incoming chemical signal. Then it dissolves in this yellow part. That's the chemical, where the mucus breaks down the chemical. And then it's basically able to come and do this. It triggers another G protein. Then it goes to adenylene cyclase, and then CAMP uses, it's a ligand. Let me change that. That's a ligand gated receptor channel. Cool. And so smell is the only major sense that doesn't go to the thalamus. It goes to the temporal lobe, which means it gives us acute memories. The reason it does that is because if you can kind of think back to when you're a caveman, right? If you smelled fire, you knew danger. Or if you smelled good food, then you need to go to it. So it goes back to survival instincts. But that's why it's like a bigger deal to wear cologne and stuff than you think it is when you're trying to court a woman, or vice versa, when a woman is trying to court a man, or whatever way you swing. Okay, finally, here's vision. We'll go through the anatomy real quick. This is the cornea right here. The cornea is the outer layer, the front layer of the eye. And this is the pupil. And what causes the pupil to dilate and expand are the iris muscles, which are the ones that give you color. My eyes are blue, so I did that to kind of remind me of that. Inside of that is the aqueous humor. That's it's kind of like a gel-like substance. And then we have our lens here, suspensory ligaments, and ciliary body. And so if we contract that, you can think of it kind of like a sphincter like this, and we contract it, it would make it smaller, which would then release tension off of those suspensory ligaments. When it releases tension off of those suspensory ligaments, it makes it more concave, like so, more like the shape of a football. Light will then, light will then touch it, and then it is able to go like this. So it goes like that and focuses in. Versus the other way, if we want it to go more far-sighted, we're gonna get light. It will hit the cornea and refract just a little bit. It'll go in, and then if we want that, then it'll keep going straight. If we just flatten it, we would then relax those muscles. We'll talk more about that in a little bit. We have right here, I haven't drawn it, it's the fovea. That's a high concentration of cones. That's actually where you're trying to focus with the lens, because it gives the greatest acuity. And the reason it's able to do that is cones, the retina is made up of cones and rods. Cones are able to, they're like black and white, and they're able to pick up vision pretty well. And cones are give you that acute sense of seeing and color. And so what happens is that fovea is full of cones, and the rest around the side is full of rods. And for every, let's say like 5 rods, there's 1 receptor. So I have all these like this, and they all go to the same receptor. Versus our cone, it'll go like this, and then I have 1. So I'm able to pinpoint and kind of more accurately how the eye is able to, how it's able to work, or how it's able to pinpoint things with cones better, because it's more specialized and more specific. So that's kind of how that works. The sclera, that's the white part, makes up most of the outside of the eyeball. The middle eye has lots of veins. And we've kind of talked about this. Going here to lens, we talked about the ciliary muscle, how that works, the concaveness. Basically, we have hyperphobia and myophobia. That might be wrong, but you'll get the point. What happens is either our eye is too short in this one, or it's too long in this one. And what that causes is it doesn't have a chance to really pinpoint early enough. So we're gonna need some glasses that would magnify, or they'd be concave, they'd push the light like this better. Versus this one, it's coming together too early. We need it to be together right there. So we need glasses that'll make this more flat, make it go more straight so that it hits the end at the right spot. Okay, now for the good stuff. How does the actual signals work? We talked about the cones and the rods in the back of the retina. What will happen is we're going to do this for a rod. Cones are similar. Just remember that they are more of a 1-to-1 ratio. So right here, it'd be just like this instead of those. There's some other differences, but we're not going to focus on those. So what we have is rhodopsin right here. It's made up of 7 opsin around the retinal. The retinal then receives light. It changes to a more stable one, which is trans. Trans is always more stable than cis, so that's why it's going to do that. When it does that, it's going to activate this rhodopsin. It'll pick up this transducin. This will come here, as you see right there, and this will activate the PDE, which you can see is in green. This will then take the cyclic GMP, break it down, and the GMP will then block these channels so the ions can't come through. So anytime a cell is depolarizing, it's because it's dark. Light causes less glutamate. Less. Light, less. Okay? So always remember that. What deactivates that rhodopsin is an arrestin, okay? So coming down here, we have what are called on cells and off cells. These are the bipolars. These are photoreceptors, bipolars, and ganglions, okay? So if I have an on cell, that means it has what's called a little mGluR6 right there, which when this cell is spontaneously depolarizing and sending action potentials. When I add acetylcholine right here, it will then come and block off the TRPM1, which is not good. So this is why it's good to have light. As light comes, it makes little to no glutamate. And we have something right here, it's the horizontal cell. That gets the glutamate, activates that. That in turn releases GABA, which lets less glutamate through here, which means less blockage of our TRPM1. And so that is good for getting more signals. In our off cells, it's a lot more simple. They respond better to dark because they need glutamate to activate the AMPA, which then depolarizes, sends a signal down, and we're on our way. We have glutamate right here, sending an action potential down the synapse. Okay, something that's cool here is in between the rods and the cones that are on the retina, we have something called pigmented epithelium, which absorbs the light, and it takes the all-trans retinal that we had, and it takes it back to cis. So then it kind of resets those cells. So those are really important. Okay, last thing, we have hearing. Hearing works like this. We have an ear canal. It goes in the ear canal. We have a tympanic membrane. What happens is we have pressurized air that comes in. The air then touches this. We have a bony structure that amplifies the noise. So it amplifies the noise by then sending a fluid down this, which will then go all the way around and then back out the other side, like that. And so we have, this is it kind of blown up where it goes. This is kind of if I took that whole thing, cut it in half, and now I'm going to take even a smaller one of those and put it right here. So this is where it goes in, that the auditory ossicles send that action in, and this is where it comes out. And what's going to happen is we're going to have fluid that's going to vibrate this right here. And so when it vibrates on this tectorial membrane, if it vibrates down here, that's our high noises, high pitches, and down here is low pitches. And so it actually rattles easier up here. So we're able to easily hear, more easily hear low tones. And so it's more stiff right here. But what causes is our hair cells are become more damaged right here as we go through our life. So when we get older, we have a hard time hearing these pitches. And so if you ever have a hard time talking to an old person, try talking to them in a lower tone. You might be able to reach through to them. Okay. This is full of perilymph fluid. This part right here, it says right there, you can hardly see it. So is this, and then this one has something called endolymph fluid, which is made by the stria vascularis. The stria vascularis works like this. It's, I don't have the diagram, but it's secondary active transport that's pumping potassium all the way through. And that creates an extremely strong gradient. And so we have an 80 millivolt driving force to get through right there. And then we have another 40 to get through to here. So then in turn, its potassium has a 120 millivolt driving force to get out of there. And so how does it let it in? What's going to happen is those vibrations are going to put the endolymph fluid in here, right? And then these outer hair cells are going to churn it up and down, forcing it to go in there. What that's going to cause is it's going to cause these to push over like that. When they push over, they have these ligand receptors that have tip links. Those tip links are going to come open, and the potassium is going to come through along with a little calcium, and it's just going to shoot through this thing, okay? And then it'll release glutamate and send an action potential that way. Now, the outer hair cells are extremely important because they are regulatory. Those are efferent signals. Our brain we don't pay attention to this, but it will send signals so that we can tune people out or listen more closely. And how that works is these will either lower or raise. And so if the closer they are to the tectorial membrane, the better we are going to hear. And so if we fall asleep in a room and there's tons of people talking, these are going to come down, and there's going to be less endolymph knocking this inner one over, which means we're going to be able to hear less. Versus if you hear your name in a crowd, these are going to come up, we're going to have amplified hearing, and it'll work out a little better. So that's pretty cool. Let's see. Okay, there's 3 types of hearing loss. We have sensory neural, which is kind of the way I was talking about. The older you get, those hair cells become damaged. So you just slowly lose your hearing. We also have central hearing loss. That comes from like a stroke or something, that where your auditory cortex is, you're not able to process the sound either at all or in not as good as a way. And then we have a conductive blockage. Conductive blockage means we have like an infection or earwax that's on the outer ear that's blocking us from hearing well. And then we have balance. Balance, I like to think of this one as vestibule. If a vestibule is like between like when you're on campus, it's, okay, you're walking in the doors of the Benson, okay? And there's that little room that you don't really know why it's there, and you're pretty sure it's there just to keep temperature from, flowing and stuff. It's like, also it helps with drafts. So that's a little vestibule, okay? And so I think of that as you never really stop in the vestibule. You just keep walking through. So it's like linear. You don't stop or turn or anything. You just walk straight through a vestibule. And then so that's responsible for your linear movement. Those are, that gives you balance and linear actions. Then there's semicircular canals, the angular rotational movements. That'd be bobbing my head up and down, left to right, to just like tiltations and stuff. You can kind of think that semicircular is angular, right? It goes, or rotational. Circles rotate, so it's semicircular. So that's just how you can remember a few of those things. I think that's covering everything. I'm just going to look through real quick how energy is carried. Yep. The eyes, smell and taste. Cool stuff. All right.